on antiretroviral therapy and subsequently died. So we have a lot of tissue from other, in fact, every organ, and we're going to do some sort of specialized sort of staining in there, but nothing like flow and trying, because you start dealing with really limited specimens, and lung lavage fluid is messy enough, and you actually get quite a bit of cells, but it's still pretty messy. So I think we need some new tools there, and I think some of them are being developed, and I've been keeping my ears open, but no, not yet. Yes?
Can you demonstrate the correlation between pulmonary and peripheral blood immune parameters? Like in these peripheral blood samples, so don't really characterize what's going on in the lung.
That would be the hope. I mean, so I after seeing a couple of panels like that and patients like that, I would hope that there would be a correlation between the two, and that because obviously doing lung lavage on everybody is not going to be ever feasible. What I have seen is that in general, yes, they do correlate. When you get a broad response in the lung and the blood, there is quite a bit of cytokine frequency and within the CD4s and CD8s as well. So there is a general correlation there, but again, it's very limited thus far. So yes.
Sort of if the patient you showed had such unbelievably robust lung-specific responses, it's extraordinary. So there is some data on, I think, induced sputum as a source of, you know, it's not like a lung lavage,
Right.
but with robust responses like that, can you conceive of induced sputum being a substrate for flow cytometry that might be able to distinguish iris?
We've tried that too in another study. Unfortunately, the induced sputums are quite variable. It's possible. I mean, you can actually, if you get a proper induced sputum and you let it go full on 20 to 30 minutes of nebulization, you can actually start sampling the alveolar space. But it actually takes a good 15 solid minutes to kind of start sampling where you think you should be, and the quality is quite variable. You have to ask yourself, why haven't his induced sputum ever really taken off? It's theoretically easy to perform, but it is variable and it is kind of a pain, and it's time consuming. So I would hope so, and I think, if we could get to that point, it shouldn't be more justification to try to employ it more often. But we've tried some of that as well.
Did you have TH17 in your polyfunctional panel?
No, don't have TH. No. Those aren't in the flow panels now.
I thought you had shown that before, that you were going to include TH17.
So we're going to actually, the third aim is actually to harvest all the supername. We're going to do multiplex cytokines, so that'll be actually give us the additional data on the TH17 and TH2 balance as well. There's only so much we can stick in a flow panel, but.
And can you get PPD-induced TH17 as an antigen-specific response?
That I don't know.
Okay. Thank you. Can I just pause and ask, does anybody have a feature that's not quite so,
Yeah.
well, that turns off?
Turn it off. Can I see?
Yeah. Oh, I actually did.
Yeah.
Hold on.
Yeah, there's another button.
Now it's, yeah.
That's a
That
green.
was the one I was pressing.
There you go.
Okay.
All right.
So it goes
All
on.
it took, oh,
Okay.
thank you.
There you go.
Okay.
Thank you. So it's this button.
Okay.
Okay, so it is my pleasure to introduce Jinhee Lee, who's from the University of Massachusetts Medical Center, who's going to talk about the potential use of humanized mice as a model for HIV-TB co-infection, or TB, starting with the TB from the TB perspective.
Yep. Thank you, Siva and especially Sarah, for inviting me. But I don't know why I'm here, because my talk doesn't have any HIV components. But I strongly, strongly encourage you to think about how to use this human mice for HIV-TB co-infection. Today, I'm going to talk about my recent progress in development of humanized mice for TB research. In many research disciplines, the humanized mice is proposed as a jigsaw puzzle block able to bridge the gap between clinical studies and clinical trials. So and the humanoid mice are also bear great potential to be a fascinating animal to study human immunity to TB. So despite the negative trends about murine models, there are many good reasons to use the mice, including the unbeatable convenience. But mouse are not humans. There are many critical differences that limit the interpretation of murine data. That includes mouse don't have latent TB, and all the mice succumb to primary TB, which is rare in humans, except for HIV patients. And mouse do not develop granuloma, which is the hallmark of human TB. So if we can regenerate a human immune system in a convenient mouse system, and by injecting human hematopoietic stem cells, and this the human system, if the human system developed granuloma and TB resistant, just like humans, and latent TB, they will revolutionize translation of preclinical studies to clinical trials. But human cells cannot survive in immunocompetent mice because of xenoreactive immune responses to human cells. So the mouse, it is essential to generate immunodeficient host mice to better support human cell development. So a series of breakthroughs have been made. They include SCID mice, severe combined immunodeficiency mice, and a combined SCID NOD combination. And lastly, the generation of IL-2 receptor gamma knockout mice. And SG mice which combines NOD, SCID, and IL-2 receptor gamma null traits are the most advanced form of immunodeficient mice that for engraftment of human cell development. Equally important is the developing technologies for human cell engraftment. The earlier SCID mice were injected with intravenously with PBMC and human hematopoietic stem cells, but the rate of engraftment is very low, and there are very few T cells and no functions. The latest and the most advanced form of human cell engraftment, the BLT method. BLT stands for bone marrow and liver and thymus. So here's how it is BLT method generated. The sliced small fragments of human fetal liver and fetal thymus implanted under the renal capsule of NSG mice to make a thymic organoid. These mice are sublethally irradiated and injected intravenously with the CD34 pipes from the same fetal liver source. So this BLT mice supports the almost full array of human immune cells, including CD4 cells, CD3, CD8 T cells, and even Tregs. Like this way. And dendritic cells and monocyte macrophages. And of note, this BLT mice permits HIV infection through the vaginal route, because there's the human cells, the presence of human cells in the mucosa. So this is my only reference to the HIV. So no more HIV in my talk. So I took these BLT mice and looked at human immune responses in mouse system to BCG. First I will show you data indicating that human adaptive immune response is BCG infection. First BLT mice are infected with BCG intravenously. 1 month later, the splints are collected to measure human T cell recovery responses by ELISpot and ELISA. The result from ELISpot assay clearly shows that human antigen-specific human T cells are primed, and they demonstrated memory responses to ex vivo stimulation with various MTB antigens, including culture-filtered antigen and heat-killed BCG whole bacteria. So it may not be that much, but this is really big deal for me. So I was quite excited, and this the detection of adaptive antigen-specific T cell responses marked the major advancement of development of humanoid mice for TB research. Interferon ELISA result also shows that antigen-specific T cell responses here, but as you see, the levels of secreted interferon gamma was very low, just barely over the baseline. This is the problems that we have to solve. Regarding immunohistochemistry, this HNE staining shows that BLT mice develop lesions in the lung of BLT mice, which are comparable in size, but the fewer in numbers compared to those in C57 BL6 mice that are parallelly infected with the BCG. NS mice have smaller and the fewer lesions. To see if these lesions are granuloma found in the BLT, serial sections were stained with exit-fast and human macrophages and human CD4 CD8 T cells. These BCG bacilli are found in all of the lesions, but they are also found in outside of lesions. And human macrophages stained with human CD8 is very abundant in the lesion, and they are all evenly distributed. There are significant recruitment of CD4 and CD8 T cells in the lesion, but the frequency is quite low, and they don't, not like granuloma, they don't form a cuff to surrounding these macrophage lesions. So these lesions are not quite granuloma, but it's suggestive of induction of innate immune responses and adaptive immune responses. So I got some adaptive immune responses. So I'd like to test whether this induction of adaptive immune responses can be translated into protection, which is reduction of bacterial load in the lung. So first of all, I want to tell you that NSG mice are susceptible to BCG. So they allow continuous BCG growth until death. So the question is, could these BLT mice can control BCG growth with the demonstrated adaptive immune responses? To my disappointment, there's no reduction in the lung, the reduction of bacterial load in the lung of BLT mice compared to NSG mice. Even I should say there are more BCG bacilli in BLT mice. It's quite confusing. So I was a question why human immune responses failed to provide protection, and why there was higher bacterial load in BLT mice. First thing that came to my mind is that mouse macrophages, because BLT mice still have residual mouse macrophages. So I hypothesized that residual murine macrophages are dominant macrophages harboring BCG bacilli in BLT mice, which prevents effector human T cells from controlling BCG growth due to species barriers. To test the hypothesis, I measure first relative numbers of murine and human macrophages by staining with urine F480 antibody and human CD68. Without infection, there's comparable frequencies of murine and human macrophages in the lung. But in the lesion, there's fundamental differences in the lung. Immunostaining of serial section of the same lesion visualized by DEPI staining. Human macrophages constitute the most macrophage population for the climax is on the next slide. Sorry. when sections were stained with anti-PPD to visualize BCG, the lesions that contain the most concentrated abundant BCG bacilli compared to non-lesions area. So this, so think about, remember that these lesions are mostly composed of human macrophages. So here's what I think happened. The human macrophages are recruited to the site of infection and form a macrophage mass and serve as a greenhouse for BCG growth. And which is the typical immune responses in both murine and human TB. When you see BS6, 15 BS6 mice that are parallelly infected with BCG, there are a lot fewer BCG bacilli. It's hard to see the BCG bacilli in the region of C15 BS6 mice because these mice have efficient effector T cell functions. So these BLT mice have early responses like macrophage recruitment, but lacks efficient effector T cell functions. And when you look at, if you look at NSG mice, there are a lot less macrophage recruitments to fuel the BCG growth. So when you compare these NSG and BLT mice, you can see why there are more BCG bacilli in BLT mice than in NSG mice. So in conclusion, the hypothesis is rejected because human macrophages are dominant macrophages in the region. And why the human immune response failed to provide protection is because of inefficient human T cell response in the region. And probably low number of T cells in the region and then poor interferon production. But why there are high BCG load in BLT mice? Because human macrophages form a macrophage mass to fuel BCG growth. So the future study should be directed to identify ways to enhance effector T cell functions in the region. So this Hardy Confield is my mentor. He's a tremendous support for my research and career. And most importantly, he let me do this project. His lab members helped me in many ways. Dale Greiner and Mike Bram in medical school, and Lenny Schultz in Jackson Laboratory. This trio leading the humanized mice research provide me with all the BLT mice and all the critical discussions. Thank you.
Thanks for the talk. Just a question for you on your talks about some of the reasons why these granulomas may be failing to control TB infection. I'm wondering, if you think about developing the humanized mouse model, how much does the role of sort of the non-immune parts, like the lung architecture, the non-immune lung cells, play a part in granuloma functioning? Can we ever sort of perfect that?
I read a paper that because of a small size and I don't know the term, because of the mechanical things, so the mouse never can develop a granuloma. But that's I don't know if that's true or not. But I think when you see the lesions, there's a very the lesions are a lot closer to human granuloma than mouse lesions because which are very the structure is very unorganized. But this structure in the lesion found in BLT mice have some kind of the structure. So I think I will see whether if I can manage to increase the effector T cell functions and there are more T cells, you will see how it happens.
I'm wondering, in your system, do you know if the human macrophages in the BLT mouse are making nitric oxide?
I haven't measured that. That's a very good question.
Yeah,
Thanks.
oh, time's up,
Yeah.
sorry. Enough of that. Policy leader.
Yeah.
Thank you very much. Okay. So next we have Judd Walson from the University of Washington, who will talk about the prevalence and correlates of tuberculosis identified in non-pulmonary specimens from severely ill hospitalized children in Kenya. And you have our sort of deadly pointer there.
This looks like a clumsy pointer.
Yeah.
Well, thank you very much. It's really a pleasure to be here, and thanks to the organizers for putting this together. I really first want to just speak about our CFAR at the University of Washington. We do have a scientific working group called AIDS, or the AIDS-Associated Infections and Malignancies Scientific Working Group, which we've now proposed to be morphed into a core in our renewal application. And through that working group, of which Corey Casper and myself co-directed, we really are trying to focus on increasing sort of recognition and work around TB HIV co-infection in Seattle. And that's something that we're opening up to a lot of institutions in the Seattle area, not just the University of Washington, but other members like the Fred Hutchinson Cancer Research Institute and Gates Foundation, Seattle Biomedical, et cetera. And so I'm really hopeful that through collaboration and through meeting all of you, we're able to continue growing that. So I think one of the main goals that we have as a group is really reaching out to investigators who may not traditionally have been involved in HIV research and trying to bring them into the CFAR. And that's really been a main focus. And so reaching out to people who've been working in traditional tuberculosis research and getting them involved in TB, HIV. And also, I think, trying to leverage the expertise and knowledge of people in the HIV community to try to share some of their methods, et cetera, and maybe apply them in a new way to TB and HIV. So with that said, I want to talk a little bit about some work we're doing around some work in Kenya. I guess I have to stand over here. So first of all, I'm not going to go into too much detail here, only to highlight the fact that although a substantial proportion of tuberculosis cases, incident cases around the world occur in children, we don't actually focus on them in many programs for a variety of reasons, mostly because from a public health perspective, they don't pose often a huge infectious risk. So both the diagnostic modalities that have traditionally been used to identify TB in children, as well as the focus in terms of trying to get them on treatment, has been reduced because of those pressing public health issues. But it doesn't mean that there isn't a substantial burden of TB among these children. And so we're going to talk a little bit about looking at this among severely ill children. So again, when we look at what are risk factors for having non-pulmonary tuberculosis or extrathoracic TB, I think it's notable that really age being less than 5 is a big one, and then malnutrition and immunosuppression. And of course, if you spend any time in high HIV prevalent areas in sub-Saharan Africa, these three things many times occur together. And so part of the question is how many of these severely malnourished HIV-infected young children who we're commonly seeing with sepsis-like syndromes in the hospital may be actually suffering from disseminated forms of tuberculosis. So again, this is an educated audience. I'm not going to spend a lot of time on these, only to say that there are a variety of new diagnostic modalities that are now coming online that you are aware of, but their role and their place in the diagnostics for children is still not entirely clear. And so part of what we're hoping to do is trying to decide whether there are better or more optimal ways to diagnose tuberculosis in young children. And these are just some data which I'm sure many of you are familiar with, looking at the use of the gene expert for the diagnosis of pulmonary tuberculosis in Africa and showing incredibly high sensitivity and specificity for pulmonary TB. And this is another diagnostic, the urinary lamb assay. There are several constraints to this assay which you're aware of, I'm sure, in terms of its sensitivity. But I think, as many of you are also aware, the more immunosuppressed you are, and also in terms of renal clearance, there are some issues that may actually impact the sensitivity of the assay. And young children, for a variety of reasons, may have very differing results to the urinary lamb testing, both because of differences in their GFR and just innately because they're young children, and as well as their relative level of immunocompetence, both due to their young childhood nature as well as their HIV disease. So there have been a variety of studies that have looked at severely ill adults and looked to see whether or not circulating tuberculosis can be detected in the blood of severely ill patients. Now, I'm not going to go too much into this because the last talk of today is Shevin Jacob, who the first study listed there he's, I'm sure, going to talk about, so I don't want to go through that too much. But I would say there have been several studies which have showed shockingly high rates of circulating tuberculosis in the blood of adults with severe illness in Africa. And there have been a few studies which have not shown that. And so I don't know that it's completely sorted out, but I think this is compelling and interesting, suggesting that, in fact, severe illness, there may be a mycobacteremia syndrome associated with severe illness. In children, there are far, far less data available to draw from. There have been a couple of small studies, and neither of these studies have shown high rates of circulating TB in the blood of children. And this is really important because the actual disease itself may be very different in children. It may not initiate from a pulmonary focus with continued bacillary dropping from a pulmonary focus. It may actually represent primary infection with a big dissemination event, but that temporally may be harder to detect at one point in time. So I just want to quickly, I don't have a lot of time left, but we are doing a variety of studies in Kenya, both in Nairobi and in Western Kenya, that we're going to leverage. The first study is a U19 award that I'm a project PI on, looking actually at the emergence of non-typhoidal salmonella as a cause of sepsis. But we're using these cohorts to be able to evaluate among suspected sepsis, children with suspected sepsis, whether we're able to document the presence of MTB in these children. And here is the specific aim related to that. And we'll be using both liquid culture as well as urinary lamb, and we're in the process of trying to see if we can use stool for a gene expert as well in these children to see just exactly how much we can detect in terms of circulating mycobacteremia in these young children. And this is the children that we're proposing to do. So these are sick children with fever who are admitted to the hospital, essentially, in Kenya. These are all of the data that we're collecting in addition to the laboratory testing I described, just so you can see there's a lot of information being collected that we can then go back and look and make some associations with their risk of TB infection in terms of demographics and malnutrition status, etc. And then we have another study going on to look at the early initiation of antiretroviral therapy in severely ill children in Kenya. This is being done in Nairobi. The primary aims of that study are really to look and see whether initiating ART immediately is of benefit compared to a slight delay. And within that, we are going to look at the diagnostic performance of a whole combination of TB tests in this cohort to determine exactly among severely ill children with HIV in the hospital prior to antiretroviral therapy initiation, how many of them have evidence of tuberculosis and whether or not some of these tests perform better than others or in combination. And so here again is the study population. We'll be looking at 300 of those kids in an attempt to document that. And those children also have quite a lot of data being collected on them that we will be able to look back at after we've collected these laboratory assays. So I think the main points of this are really to see if we can develop a series of algorithms that employ more rapid and sensitive diagnostics for TB in children, understand the TB pathogenesis of disseminated TB in this group, and really I think we're hoping to get some pilot data to establish further grant applications in this area. And I think from a clinical standpoint, we all know who've worked in Africa that many severely ill children, if they're not better by day 2, are not dead by day 2. And in our studies, the severely ill children have about a 50% chance of dying within the first 48 hours after admission if they're malnourished and severely ill. So those that survive are often started empirically on anti-tuberculosis therapy pretty quickly. And then they're continued on. And we don't know how many of them actually have TB, but